Patterson here. Today we're going to go over cellular respiration. This is AP Biology, Unit 3. This is a pretty complex topic, so this is going to take a little bit of time. You guys like to get this cellular respiration and parts of photosynthesis mixed up. There's some similar pieces to it, but just take your time, go through it, you'll get it. So cellular respiration is the breakdown of chemical bonds in sugar to release energy in the form of ATP. So if you remember, in photosynthesis, we were building sugars. I had to get all my C's, my H's, and my O's to make sugar. So now I have sugar, I have glucose, and I got to break all these bonds down. And I can't do it all at once. If I broke down all of these bonds at once, I would release so much energy. Things would catch on fire, things would explode, it would create mass chaos. So we don't want that. So we need to have it break down in small little chunks, little intervals. So this is what cellular respiration has to do. So we start with different parts. We have what's called glycolysis. I have pyruvate oxidation. I have the Krebs cycle, and I'm going to end over here with the ETC. So all of that is going to produce a bunch of energy that I can use to run, to do daily metabolism. I'm able to use this energy for that. So we have to talk about what's called a redox reaction. And if you watched my photosynthesis video, we kind of talked about this. So these are when electrons are transferred from one reaction to another. I have what's called an oxidation. This is where I lose electrons. I'm going to lose one or more electrons from a reaction. And if I am reducing or have a reduction, I am gaining. You guys want to tell me that if I'm reducing, I'm losing electrons, but I'm actually gaining. Another atom is losing electrons. I'm gaining one or more electrons from another molecule. So we use this example. Leo the lion says grr. If I lose electrons, I oxidize. Gain electrons, I reduce. So here I have a formula. I have glucose, I have oxygen gas, carbon dioxide, water, and energy. And that energy is going to come in the form of ATP and heat. So this is my cellular respiration formula. I'm going to have oxidation and reduction. So remember, if I'm oxidizing, I'm losing electrons. I have sugar here, and I have carbon dioxide here. Did I gain or did I lose electrons when I'm coming to carbon dioxide? Look at what I've done. I've lost a lot of stuff. So if I'm losing electrons, what do we call that? It's an oxidation. Now let's look at the other part of my formula. I have oxygen and I come over here to water. I had oxygen and now I have hydrogens attached to that. So if I'm gaining electrons, I've gained some stuff over here. What do we call that? A reduction. So I am reducing oxygen to water. I am oxidizing sugar into carbon dioxide. What formula does this remind you of? This reminds you of one we just kind of went over, right? So it reminds you of photosynthesis. So photosynthesis, I took carbon dioxide and water, and I took energy from sunlight, and I made sugar and oxygen. So I had a reduction. This here gained electrons, and then this was reduced. Okay, water got oxidized into oxygen gas. So this was reduced. This is oxidized. So let's go back to our cellular respiration formula. I have my sugar and oxygen, my carbon dioxide, water, and energy are my products of this reaction. So this is an exergonic release of energy from glucose, and it's used to phosphorylate ADP to ATP. Do you remember what it means to phosphorylate? I'm going to add some phosphates to this ATP. Remember, I had 2 phosphates here. Now I'm going to have 3, so I'm adding a phosphate to this, and this happens with cellular respiration. I need to harvest energy, and I can harvest energy by breaking down glucose, and I got to do it in tiny little steps. Remember, I can't break them all down at once. I've got to break it down in small little chunks. So this is an exergonic pathway. Remember, exergonic means energy is released. So I'm going to break it down with glucose. I'm going to have NADH. I'm going to have an ETC. And oxygen is going to help pull this is going to be my electron acceptor. This is going to help keep this machine going to allow energy to come out. So we got to harvest these electrons. So electrons are stripped from the glucose, and each electron taken travels with a proton. Remember, those are those hydrogen protons. So I have no electrons, it's just that proton. Dehydrogenase, remember, if it ends in ASE, what does that mean? It's an enzyme, right? So this is an enzyme. Remember, it's a protein wrapped a certain way. If I change my shape, I change my function. This is an enzyme. So kind of have that in mind when we're talking about enzymes. So this is going to take 2 electrons and 2 protons from my glucose molecule. It's an oxidizing agent for glucose. Remember, what does it mean to oxidize? Leo the lion says grr. I'm going to lose electrons. And what did I take from it? I lost 2 electrons. So this is going to oxidize glucose. I'm going to lose some electrons from that glucose. So here's my glucose molecule. I'm going to have these H's are going to get pulled off. 2 hydrogen protons are going to get pulled off. And then I'm going to have 2 electrons are going to get pulled in too. So it's going to rip those pieces off or take them off this glucose molecule. Then I'm going to have these 2 electrons and 1 proton is going to get passed to our electron carrier. Remember, in photosynthesis, we had something very similar to this. You remember what that was called? That was NADP+. Okay, so right now in cellular respiration, that P is not there. I have NAD+, but this works the exact same way. This is the taxi. I'm picking up a hydrogen and I'm picking up 2 electrons, and I'm carrying this to the ETC. So I'm going to have NAD+. It's going to get reduced. Remember, if it's reduced, Leo the lion says grr. I gain electrons, I'm reduced. This is going to get reduced. So I'm going to take one of those hydrogens, are going to get attached. The other hydrogen is going to get put out into the solution, which we're going to talk about why this is important later. And then these electrons are going to go onto this NAD+. So now this has now been reduced to NADH. So I have the hydrogen and 2 electrons. This is now going to move to the ETC. All right, so that's heading out. So the ETC is your electron transport chain. Remember, we saw one of these in photosynthesis, but it looked a little different. Remember, you had photosystem 2, you had your ETC, photosystem 1. Here in cellular respiration, I have complexes, and you can see I have complex 1, 2, 3, and 4. And these work kind of the same way. I have electrons getting passed through this ETC. As it does, is going to create a charge, and I'm going to create hydrogen pumps. This electric negative pull is going to pull these positive protons through this pump, and I'm going to create a huge concentration gradient over here. I'm going to build hydrogens over here. Remember, they don't want to go there naturally. They have to be forced. And this negative charge is going to attract these hydrogen protons and pull them through. So where are these hydrogens and these electrons coming from? Our friends, NADH, this hydrogen is going to get pulled off. My electrons are going to get pulled into this ETC, into complex 1. And then I'm going to turn into NAD+. And this NAD+ is going to go back to glycolysis. It's going to go back to that area for me to get more H's pulled back on. We have another electron carrier. This is a new guy. His name is FADH2. FADH2 is grabbing 2 hydrogens and 2 electrons. And now I'm going to come to complex 2. So NADH comes to 1. FADH comes to 2. FADH is going to pull off hydrogens, and it's going to pull off some more electrons. Electrons are going to come in here too, And now I have electrons coming through complex 1, electrons through complex 2. And these electrons are going to get pushed through this ETC, causing a negative charge that's going to have all of these hydrogens start to come and fill up this membrane out here. Okay, it's going to release energy to make ATP, because remember, just like in photosynthesis, this concentration of hydrogen protons is going to run ATP synthase, and this is where I make my ATP. So transfer of electrons to oxygen. So I'm going to have oxygen gas down here is going to make water. So oxygen is my final electron acceptor. Oxygen has to come here to the end of this ETC to pull off these electrons. As it's pulling off these electrons, it's going to grab 2 hydrogens that are here, and I'm going to have water that's made as a waste. So when you guys are exhaling, if you had glass or a mirror in front of your face, it's going to start to condense with fog. And it's the water that is being made from this process that's going to come out. So oxygen gas is going to be here. It's going to pull an electron off. It's going to pull some hydrogens, and I'm going to have water. If this oxygen is not here, and this is so important to understand, if this oxygen is not here, this whole ETC shuts down. And if this ETC shuts down, what else shuts down? Pyruvate, okay, glycolysis, all of that starts to shut down. So this is super important that this oxygen is here as a electron acceptor. I've got to get this oxygen. This is also why if you don't have enough oxygen, all of a sudden stuff starts shutting down, because this process isn't allowed to work to make ATP. And we're going to get more in depth in this here in a second. All right, so I have 3 stages. One is a half stage, and this first stage we're going to talk about is glycolysis. So glycolysis is when I'm going to make 2-pyruvate. This happens outside of the mitochondria. So it's going to start in the cytosol. It's going to start breaking down glucose into 2 molecules of pyruvate. So I'm starting my small breaking down pieces of the sugar. So here I have glucose. I have a 6-carbon sugar, 1, 2, 3, 4, 5, 6. And I'm going to take it, and I have to take ATP. Remember, this isn't going to want to break down on its own. So I have to start the process. So ATP is going to come in, and I'm going to have 2 ADP come out. But this is going to break down this 6-carbon sugar into 2 3-carbon molecules. These 3-carbon molecules are then going to have hydrogens harvested off. NAD plus is going to come in. It's going to start pulling off hydrogens, and it's going to pull off some extra. And then ADP is going to come in, and then I can actually make some ATP in this process with a sub-level phosphorylation. It's going to come in and make some ATP. That is going to make 1 pyruvate, and I need this for the next cycle. I need this pyruvate. But remember, I had 6 carbons. This was just 3. So what do you think is happening over here? The exact same thing. So 1 glucose in glycolysis, I can have NADH, I can have hydrogens, and I can bring in 4 ATP. So they call this part here my energy investment stage. I had to invest 2 ATP to get this process started. Once this gets put in, then it just kind of starts to work. So this ATP is breaking this 6 sugar down into 2 3-carbon molecules. I'm making NADH. I have hydrogens coming in. NADH. I have some hydrogens. And then ATP over here. So I had to invest 2 ATP to make 4 ATP. Okay, so I have 4 over here. And then I have 2 NADs that are going to make 2 NADHs, and I'm going to have 2 extra hydrogens left over. So my net is 2 pyruvate, 1, 2. I'm going to have 2 water molecules that are going to come out of it. But look at this. I'm only netting 2 ATP, but I made 4. I made 4 ATPs. So why is it saying I only have 2? Remember, I had to pull 2 from here. So I had to have 2 ATP to start out, and then I made 4. So it'd be like having $2. Okay, I got to pay $2 to make $4. So I have to take $2 out of that. So I'm only going to have a profit of 2. And that's kind of what this is saying. I have a net profit of 2 ATP. I'm going to have 2 NADHs and 2 hydrogen protons left over. So this is what I'm going to get just for glycolysis. I do not need oxygen for glycolysis to happen, okay? So now I have glucose has come in. I've gone through glycolysis. I've made 2 pyruvate. I need these pyruvates for the next step. Something else I made was 2 ATP, so that's going to come out of the process. And then I made 2 NADHs. I've harvested my H's, and I need these H's to run this ETC. So this NADH is now going to go to my ETC. All right, next step is pyruvate oxidation. So this is going to head into the mitochondria. So now I'm going to start working inside the mitochondria. So this is what they call the 1.5 bridge, or like the half step. And this is pyruvate oxidation. Remember, when something's oxidated, what is happening? You're losing electrons. Leo the lion says grr. Okay, so I'm losing electrons. So pyruvic acid is going to be converted into what's called acetyl-CoA. So here's my pyruvate. Remember, it's that 3 molecule that I made in glycolysis. Remember, I have 2 of them. This is just going to be half of that. I've got this first pyruvate I'm going to look at. I'm going to take out carbon dioxide. I'm removing one of these C's. So I'm going to take out one of those C's, and I'm going to take out some oxygen. NAD plus is going to come in. Our buddy NAD plus is going to come in and it's going to start slowly pulling out some of these H's, those yummy H's that are here on this sugar. So that NADH is going to pull that H, and I'm going to get an extra hydrogen proton in that process. And then I have to bring in what's called coenzyme A. This is an organic cofactor. This coenzyme A is now going to attach to these 2 carbons that are left. So remember, this carbon here left. I got rid of that in carbon dioxide. Now I have 2 carbons left over. I've pulled some of the hydrogens off this pyruvate. This coenzyme A is coming in, and it's going to attach to this process right here. So now I have this molecule, which is now going to head to the citric acid cycle. Okay, so that's going to head out to go to the next step. All right, so I lose one of my carbons in this process. So acetyl-CoA, I made 2. I'm going to make 2 carbon dioxides. Why do I have 2 carbon dioxides? I only made 1 in that last one. Remember, I brought in 2 pyruvates. So that process is going to happen twice. So that happened with 1 pyruvate, 1 carbon dioxide. The second pyruvate is going to give me the second carbon dioxide. I'm going to make 2 acetyl-CoAs with this. I am also going to make 2 NADHs with this. So NADH is going to head off to the ETC. Next is the Krebs cycle. Okay, so this is the Krebs cycle, or the citric acid cycle. This is going to happen in the matrix. So this is going to turn that acetyl-CoA into what's called citrate. Okay, I'm going to fully oxidize my glucose. Leo the lion says grr. I'm going to lose all of those, most of those electrons in glucose. I'm going to release all my carbons as carbon dioxide, and all my hydrogens that I can harvest are going to be gone. So this is the process for this one. I have kind of like, remember in photosynthesis, I had Ruby P. But this is going to be kind of the same thing. I have oxaloacetate. This has to be regenerated, but it's going to bond with this acetyl-CoA. So acetyl-CoA is going to come in. the coenzyme is going to leave, and I'm going to be left with these two carbons. They are going to bond to my oxaloacetate. So now I have this new molecule, and this new molecule is called citrate. The citrate is now going to get, if I'm taking off hydrogens, I'm losing hydrogens. Am I being oxidized or am I being reduced? Leo the lion says grr. So I'm going to oxidize the citrate because I'm going to lose electrons. So I'm harvesting off my H's off that. Okay, I'm pulling off a carbon. I'm pulling off some O2s, and now I'm going to have carbon dioxide waste is going to be created. So now I've lost one of those C's with carbon dioxide, and I still have some more I have to harvest. So I'm going to bring in another NAD+. I'm going to harvest off some more H's, and I'm harvesting that last carbon to make carbon dioxide. So now I have my molecules coming back, 1, 2, 3, 4, but I can make some ATP. So I'm going to be able to use this molecule, and I can phosphorylate this ADP into ATP. I can make 1 ATP in this process. I have some more H's I can harvest off this. So FAD is going to come in, and I'm going to harvest some more hydrogens off of that. I have even more I can still harvest off of this. So NAD+ is going to come in and harvest some more hydrogens on it. Once I get to this point, now I'm back to oxaloacetate. This can now come in, accept another acetyl-CoA, and start this process again. So when I have this one acetyl-CoA come in, I'm able to harvest 1, 2, 3. I'm able to make 3 NADHs. I'm able to make 1 FADH2. I'm going to make 2 carbon dioxides and 1 ATP. So remember, this happens twice. So this is the one acetyl-CoA. This is the second one because I had 2 pyruvates. I make 2 acetyl-CoAs, so I can do this process twice. So one glucose molecule, one C6H12O6, is going to net 2 ATP, 1, 2. 4 carbon dioxides, 1, 2, 3, 4. 6 NADHs, 1, 2, 3, 4, 5, 6. 2 FADH2s, 1, 2. All of this is going to be made with 1 sugar molecule. So now, glycolysis, I made 2 pyruvates, I had 2 ATPs, and I made 2 NADHs to go to the ETC. Pyruvate oxidation, I made 2 acetyl-CoAs, I have waste of 2 carbon dioxides, and I made 2 NADHs to go to the ETC. The Krebs cycle, I made 6 NADH, 2 FADH2s, and these are all going to the ETC. The Krebs cycle made 2 ATP, and I have 4 carbon dioxides as waste. I've taken out all of my carbons. Remember, I had 6 carbons for C6H12O6, and here I have 2 and 4, which give me 6. All right. Let's go to the ETC. So this is the last stage. This is going to be where I have them embedded in this intermitochondrial membrane. These are proteins that are embedded in this membrane of the mitochondria. I'm going to have 3 trans membranes. These go past and through the membrane. And I have 2 carriers. This is going to be carriers that are going to help move my electrons through this ETC. I also have ATP synthase on the other side. I have my inner membrane space and my mitochondrial matrix. So remember my mitochondria, this is the big picture of it. This is inside this membrane right here, okay? This brown part is my inner membrane space, and then this pink is the mitochondrial matrix, okay? So kind of visualize where this is at in the mitochondria. So this is going to be powered by those electrons. Remember, NADH and FADH2 brought in those electrons. And those electrons moved down this ETC, created that negative charge. This negative charge is going to pull these hydrogens through. This creates that hydrogen pump due to an electrogradient pull, okay? So at the end of the ETC, water's formed. Remember, what is my electron acceptor? What is going to be sitting here at the end of this ETC to pull off these electrons and to make water? Remember, my oxygens. Oxygen's going to sit here. I'm going to pull 2 hydrogens. Some electrons are going to get tagged onto this, and I'm going to have water get made at the end of this ETC. Oxygen is my final electron acceptor. If oxygen is not here, this whole ride stops. And if this whole ride stops, the citric acid cycle stops. The pyruvate oxidation, all of that, all of it stops. It will not allow this to work any further into the mitochondria, okay? So hydrogens are going to flow down their concentration gradient, okay? They're going to go out into this area right here. They're going to make an electrochemical gradient that is going to help phosphorylate ADP into ATP. I'm going to have a phosphate get attached to this and make ATP. So this concentration of hydrogen protons, it wants to come through. And the only way it can come through is down its concentration gradient. These hydrogens are getting pumped out. They do not want to be in this space. They want to be here. But this negative charge is forcing them into this intermembrane space. So the only way they can come back in is through ATP synthase. So they're going to come and they're just going to start flying through. As they go through this, ATP synthase is going to start to spin. And as it spins, it's going to grab an ADP, a phosphate, and it's going to slap that phosphate on to make ATP. This is where I'm going to make my bank load of ATP is here. The other ones we've been making 2. This one, I'm going to be making a lot of ATP. So this is what we call chemiosmosis. I have a hydrogen gradient across a membrane. And so this is going to create a gradient of hydrogen protons for me to go through here. So the ETC, this right here, and chemiosmosis, this hydrogen gradient, is going to result in oxidative phosphorylation. So I'm going to phosphorylate these phosphates onto ADP to make ATP. And look at the ATP I make with that. I make a lot. I'm making up to 28. So it's 26 to 28 is kind of what you're making. So here's that process again. So here I have NADH. I have NAD+. This is going to get oxidized. Okay, I'm pulling off electrons. I'm pulling off hydrogens. I'm moving through this ETC, causing a negative charge. Hydrogens are filling up this space. They're getting pumped in. FADH2 is coming in, dropping off more hydrogens and electrons to help fuel this. At the end, I'm going to have oxygen. It's going to pull off electrons. It's going to grab 2 of these hydrogens to make water. And then I'm going to have this huge concentration of hydrogen protons here. The hydrogens are going to fly through, and it's going to phosphorylate this ADP with the phosphate into ATP. So we have done a lot of stuff. I've brought in a glucose. I've gone through glycolysis. I've made 2 pyruvate and 2 ATP. Out of that, I've made 2 NADHs that have gone to the ETC to help fuel the pump. I have pyruvate oxidation, step 1.5. I made 2 acetyl-CoA. I have 2 carbon dioxides as waste. I'm making 2 NADHs to get fueled into the ETC. The Krebs cycle, I made 6 NADHs, 2 FADH2s. Those are also going to the ETC to help fuel those pumps. The Krebs cycle gave me 2 ATP, and I got rid of 4 carbons in the form of carbon dioxide. Now the ETC, I have to bring in oxygen. Remember, that's my electron acceptor. That's going to come in, and as it comes in, it's going to bond with the electrons and the hydrogens to make water. It's going to be a waste product. I'm going to be having around 26 to 28 ATP. So this is my electron acceptor. And then this is why I say like 26 to 28. Prokaryotics are going to net a total of 32. 28, 29, 30, 31, 32. Eukaryotics will net 30. And this is why. Because I have a membrane I have to go through. It does not want to go through here naturally. So 2 ATP will be used to get me inside of this mitochondria. If I am a prokaryotic cell, I do not have a membrane that I have to go into to make ATP. So that step is not there. So eukaryotics, we have that membrane we have to go through. So we take 2 ATP to make that process happen. Prokaryotics don't have that membrane, so they get a net total of 32. So now we have to talk about anaerobic respiration. So what we were talking about with this was aerobic. Aerobic means oxygen. And what was my electron acceptor for aerobic respiration? It was oxygen. So that got that to work. When we talk about anaerobic, that means no oxygen is there. So it's an electron acceptor that is not oxygen. It can be something else. So see here, I have SO4 is the sulfur, a sulfur-based molecule. This is going to come in and this is going to act as an electron acceptor. And so that will come in and it will pull electrons off this ETC. And there are some that live in areas like hot springs or in geysers where there's a lot of sulfur, a lot of hot material, and this is what they're breathing. They're breathing this sulfur molecule instead of oxygen. But they're able to do the same thing. I can make ATP, and I'm using that sulfur to keep this electron transport chain moving. So we need to talk about fermentation and glycolysis. Remember, glycolysis, we didn't need any oxygen for that to happen. So when I have glucose, remember, I had NAD plus came in, and I made 2 NADHs, and 2 ATP got made, and I made 2 pyruvate. Remember that? Okay. If I have oxygen still present, okay, I'm going to move to the ETC. And if I move to the ETC, this NADH is also going to go to the ETC. But what happens if I don't have oxygen? Remember, the ETC shuts down and then everything else behind it shuts down too. So we have what's called an anaerobic process. There are times when you can be exercising or running, and then all of a sudden when you're running, you start to breathe heavy. Okay, why are you breathing heavy? Well, your body needs more oxygen than what it's able to get. It needs more. So it's trying to get more oxygen in for these cells and muscles to be able to use all this energy that is needed. What you guys want to tell me is that this is an anaerobic respiration, and it's not. It's an anaerobic process. If it was anaerobic respiration, I would be going through glycolysis, pyruvate oxidation, citric acid cycle, the ETC, oxidative phosphorylation. But that's not what's happening here. Okay, I'm not having an electron acceptor of oxygen or some type of sulfate coming in to accept those electrons. This is just happening outside in the cytosol. This is not going into that. So this is just an anaerobic process. I don't need oxygen for this to happen. So what happens is if I have no oxygen, then that means I'm not going to the ETC, NADH can't go, it starts to fall apart. But I need to make more of this NAD+. I have a very limited supply of this. I do not have a lot. So I have to try to keep making NAD+, so I can start harvesting H's so I can make at least a little bit of ATP. I'm not going to make no 26, 28 ATP, but if I can make a couple, that's going to be enough to kind of keep me going a little bit in this process of running or stuff like that. So the only way that I can do that is to figure out a way to harvest those H's, because if not, then my NADH goes away, my NAD+ goes, and then all of a sudden, look at that, I have no more ATP, and then that's just going to be a cellular death. Okay? So we have, as animals and humans, we have what's called an animal fermentation. I need to regenerate this NAD+. So once I harvest these H's, I've got to put these H's somewhere. I can't just put them back on this. So the H's are going to go to pyruvate. That pyruvate is going to take that H and then it's going to move it into lactic acid. That lactic acid is now going to get stored in muscles and stuff like that. I have removed this H, which now gives me NAD+, which allows me to come in and harvest off some more H's to make some more ATP. And that's what I need to do. When we have plants, plants have to have an extra step. Again, I got to regenerate this NAD+. So I'm going to come in, I'm going to harvest off my H's, I'm going to make some ATP, and I'm going to make some pyruvate. But what happens now is this pyruvate, I'm going to pull off some carbon dioxide. And then that carbon dioxide is going to leave, and I'm going to have 2 acetaldehydes. The H's are going to attach to this acetaldehyde. Then this is going to make 2 ethanol molecules. And this is drinking alcohol. This is what we have in beer. This is kind of what we have in wine. And this is also why we have some carbon dioxide in those pieces too, is because of the carbon dioxide in those drinks. All right. That is it for this one. This was a long one, cellular respiration. If you guys have any questions, let me know. We'll talk to you later.